So I'm excited to give this talk. Sorry I didn't make it out there in person. At the start, I just wanted to acknowledge my co-authors. Just the work I'm going to talk about required a heroic amount of experimental effort by a number of people. Chan Lee, former postdoc, Chris Schwartz, former student and postdoc, and Kelsey McBain, who's a current technician. And then Greek Stewart is my co-PI on these projects. So we're going to be focusing on the small eukaryotic phytoplankton. And by small, I'm talking about stuff that's roughly 5 microns or less in diameter. And there's some things we know about these organisms. We know that they can be major primary producers. So especially in the open ocean, oligotrophic areas, often this small size range of eukaryotes are doing most of the primary production, or regions where Prochlorococcus is dominant, they would be like the second most important usually. We also know that they can be major predators of bacteria. So that means that at least some of them are phagotrophic mixotrophs, so doing photosynthesis, but also ingesting prey. And then another thing that I've always found really intriguing about these small eukaryotes is they have this really deep phylogenetic diversity. So molecular surveys often find all of these different classes and higher taxonomic groups: pelagophytes, haptophytes, dictyocophytes, chrysophytes, persinophytes, bolidophytes, cryptophytes, dinoflagellates, diatoms, chlorarachniophytes. So it's almost bewildering to try to think about and understand all that diversity. But one of the challenges is that they're hard to tell apart using optical methods. So microscopy or flow cytometry, they all look very similar to each other. I've just given some examples from different classes down here. And so if you think about the picocyanobacteria, obviously dominated by Prochlorococcus and Snechococcus, or you think about larger phytoplankton, tend to be dominated mostly by diatoms and dinoflagellates. There's a lot more phylogenetic diversity among these small eukaryotes, but we have much less of a handle on what the implications of that are. Basically, what are all these different things doing? So the questions that we're trying to address here is, does this deep phylogenetic diversity translate into functional diversity? Different traits, different effects on the ecosystem. How prevalent is mixotrophy? How many of these groups are doing it? And what are the trade-offs that affect, being a mixotroph? What are the grazing rates and, rates of photosynthesis? How do those trade off with each other? What are the penalties or advantages of being a mixotroph? How are communities structured across major environmental gradients? So how is all this diversity organized? And then, what are the consequences for how ecosystems operate? So I mentioned a lot of this work was done by Chris Schwartz. He's kind of a wizard at isolating things. So among the many things he's isolated from waters around Hawaii are 39 flagellate phytoplankton, I should say phytoplankton isolates. All of the ones I'm going to talk about today are from Station Aloha, which is 100 kilometers north of Oahu. And they're all 2 to 5 microns in diameter. And we've done experiments to show that all of them will eat Prochlorococcus and all of them will grow mixotrophically when you give them Prochlorococcus as the only nutrient source, so no dissolved nutrients. And then on the left here is a phylogenetic tree, which is probably a little bit hard to see, but summarizing all this diversity. So we've got 6 different classes. There's a large number of dictyocophytes in the genera Florenciella, Rhizochromulina, an undescribed genus we're calling DictyX, that's just an abbreviation. Then there's the bolidophyte Triparma. There's a clade of chrysophytes that has been described from sequence data as clade H, so I'm calling that chryso H. There's Chrysochromulina, which is a haptophyte. There's another undescribed haptophyte clade called hap2. And there's an undescribed chlorarachnophyte clade called chlor-X. And then we've got one dinoflagellate Plagiodinium. So there's a lot of diversity here that hasn't been isolated before. And for most of these groups, they've not been shown to do phagotrophy before, although it's probably suspected for a lot of them. Okay, so then one of the things we're interested in is like how good of grazers are they? So we did experiments where we fed them Prochlorococcus, and what I'm plotting here is the specific clearance rate. So that's clearance rate normalized to body volume. So, relative to your body size, how good are you at clearing prey, in this case, Prochlorococcus? It varies a lot across these different taxa. So, almost tenfold between Florenciella, which is a dictyocophyte and is a relatively slow grazer, up to this Chryso H group, which are the most voracious grazers, are the ones that we've been working with. And then there's a wide range of things in between, such as the haptophytes, other dictyocophytes, and the bolidophyte. So there's really a whole spectrum of grazing abilities here among these different taxa. And one thing we noticed when doing these experiments is that the ability to graze quickly appears to trade off with conversion efficiency. And we measured that pretty simply just as the amount of cell biovolume of the grazer created relative to how much of prey biovolume they consumed. So a better way to do this, of course, would be to have carbon-based conversion efficiency, but we don't have that data yet. This is data that's relatively easy to get. And what we find is that the species that graze faster, the isolates that graze faster, tend to have a lower conversion efficiency. So that's implying that they're respiring more of the prey biomass. So to me, this suggested that there was this spectrum of strategies here where you have the kind of slower grazers that are probably doing more photosynthesis. That's why their conversion efficiency is higher. Faster grazers that may be doing relatively more phagotrophy. That's why their conversion efficiency is lower. Okay, so to dig more into this idea of a spectrum of trophic strategies, I also wanted to add in some autotrophs, or things that we think are autotrophs. So these are 4 more station aloha isolates here. And I'm going to refer to them as autotrophs just because they don't grow when you feed them Prochlorococcus. They can't substitute dissolved nutrients with pro as prey. There's 2 flagellates, Plagomonas, which is a plagophyte, Micromonas, which is a prasinophyte. And then there's 2 non-flagellated prasinophytes, Osteococcus and Chloropecon, which is part of this commonly observed prasinophyte clade 7, which has recently been taxonomically named. Okay, so we did some more experiments to compare autotrophs and then the different mixotrophs, and to try to get a sense on what the potential trade-offs could be. So we grew a variety of strains under what I'll call phototrophic conditions. So we're giving them light and dissolved nutrients, but we're not feeding them. And we did that under a high irradiance and a low irradiance. And on the plot here, these 4 are the autotrophs and these 7 are the mixotrophs. The symbols higher are the high light and the symbols lower are the low light growth rates. And one thing that we found is that the autotrophs will grow fine under this low irradiance, 10 micromoles photons per meter squared per second. And that's pretty low. It's about 1% of the typical surface irradiance at Station Aloha, but they'll grow at 0.25 or 0.4 per day under those conditions. Compare them to the mixotrophs, 6 out of the 7 won't grow at all under that lower radiance. So that's giving us a fairly clear distinction between the autotrophs and the mixotrophs in terms of their physiology here. And then when we look at how fast these things grow under the highlight conditions, we also see that the faster grazing mixotrophs grow more slowly. So that's this correlation plot on the right here. And this one out on the end, that's that chryso H clade. It's the fastest grazer. It cannot grow at all if you don't feed it prey. And it also seems to not grow very well if you feed it prey and don't give it light. So it seems to be an obligate mixotroph. And then there's other mixotrophs that will grow, but grow slowly. So basically the picture we're getting here is consistent with trade-offs where either doing more phagotrophy means you're investing less in doing photosynthesis, or it means that you're suffering higher respiratory costs that are reducing your growth rate, or both. We can't distinguish these without further physiological data, more detailed experiments. Okay, for the next part here, we're interested then in basically community structure, like all this diversity. How does it sort of map out onto different environmental conditions? And is this spectrum from autotrophs to like more autotrophic mixotrophs to more phagotrophic mixotrophs? Does that correlate with environmental conditions? And so what we did is we took all these isolates and matched them to metabarcode data from the Terra Oceans project. So I'm plotting here on this map are 29 Terra Ocean stations at which they sampled different size fractions of the eukaryote community, including this 0.8 to 5 micron size fraction, which they called pico nano. And it's amplification based, and they were amplifying and sequencing this hypervariable V9 region of the 18S ribosomal RNA gene. And the other thing I'll say is that, it's just pretty obvious to you, but most of these are relatively oligotrophic or mesotrophic sites. So that just means that they're the kinds of environments where the smaller phytoplankton are the most important. Okay, so we have 29 isolates that we matched to OTUs, operational taxonomic units from the Terra Oceans data. And there were 13 of those that mapped onto these 29 isolates. And we're basically like matching them up with BLAST of our 18S sequences against their V9 sequences. And usually we got very close matches. And the taxonomy that the Terra group had annotated was always consistent with what we had derived from the full 18S sequences. Okay, so first let's look at what's going on with the depth. So at these stations where they size fractionated sampled the community and did metabarcoding, they had samples from what they called surface, which was like 3 to 7 meters, and samples from the deep chlorophyll maximum. And so if we compare the relative abundance of reads from our different OTUs at surface versus DCM, there's this very consistent pattern where the autotrophs are always relatively more abundant at the DCM. So this one, Micromonas, Plagomonas, Chloropecon, Ostriococcus, all those have a high relative abundance at DCM relative to the surface. And then for the mixotrophs, 8 out of 9 of them were more abundant at the surface than the DCM. So that's all these 8 up here. Then there's one mixotroph that behaves more like the autotrophs, which is interesting. And that's Florenciella, which is that slowest grazing mixotroph. And then we can get a little more quantitative comparing the grazing abilities, so the specific clearance rate when you're consuming Prochlorococcus, and compare that to the ratio of relative abundance at the surface relative to the DCM. So above this line, these are things that are, have shallower distributions. Below the lines, things that have deeper distributions. And there's a pretty strong correlation here. And I've put the autotrophs on this plot by giving them a clearance rate of 0. But you see a correlation even if you just look at the mixotrophs. So just within the mixotrophs, the faster grazing mixotrophs have shallower distributions. Okay, so that was a pattern with depth. Now we're going to look across all these different locations using the samples from the surface, those surface samples. So if we take our 13 OTUs, do a principal coordinates analysis, 46% of the variation in composition is explained by this one principal coordinate axis. And it's pretty strongly correlated with chlorophyll A, nitrate, and mixed layer depth, and has some correlation with temperature. And this is photosynthetically active radiation at the sampling depth. So basically, this looks like an axis of stratification. So you've got deeper mixed layers, higher nitrate, higher chlorophyll to the left, and then also colder and less light at the sampling depth, and then the opposite to the right. Okay, so we've got a lot of community composition that seems to be correlated with stratification, which is, of course, going to affect nutrient supply. And then we can look at where these different taxa, these different OTUs, are lining up on this axis. And so that is also correlated with grazing ability. So the faster grazers are relatively more abundant in the more stratified conditions. And the autotrophs and the more slowly grazing mixotrophs tend to be more abundant during the more deeply mixed, higher nutrient conditions. And then just to pull out one of these variables, we focused on chlorophyll A because, of course, it's very commonly measured and can be estimated from space. And so we can look at how these different OTUs vary along a chlorophyll A gradient. And this is just showing slopes of relationships estimated using a generalized linear mix model. And, you know, some of them decline as chlorophyll increases, some of them increase. And if we look at the slopes of these relationships, that's what's on the y-axis here. So this is the slope of how the OTU changes as a function of chlorophyll A concentration. And on the x-axis is that grazing ability, again, specific clearance rate. So there's a really strong relationship here where, you know, the faster grazers, they tend to decline as chlorophyll increases, and they decline more steeply. All of the autotrophs tend to increase with chlorophyll A, but even within the mixotrophs, there's a relationship. So this like faster versus slower grazing mixotrophs, there's a consistent shift in how they vary across the chlorophyll A gradient. Okay, so to summarize the empirical niche data here, we basically have two like major gradients in the ocean that seem to be leading to parallel shifts in community traits. So as you go from deeper to shallower waters, you tend to go from more phototrophic phytoplankton to more phagotrophic phytoplankton. And likewise, if you go from less stratified to more stratified surface waters, you go from more phototrophic to more phagotrophic phytoplankton. So that, of course, raises the question of what's driving this. There's a number of different things that vary across both of these gradients, two of which are nutrient supply and light availability. Of course, both of those tend to be lower. Well, nutrients tend to be lower, and light tends to be more available in the mixed layer under more stratified conditions. And the same thing happens across depths, right? So shallower depths, lower dissolved nutrient supply, but higher irradiance. So we did some relatively simple modeling to try to tease these apart. And I'm not going to go into much detail about how the model works today. But the bottom line is that, you know, modeling mixotroph competition is fairly complicated just because, you know, they're competing with the autotrophs for nutrients and for light, but they're also competing with the heterotrophs for prey. So it's like the relative abundance of these three different resources determines how the competition plays out. And on top of that, it depends on how the trade-offs in the model work, which, you know, we have some data to constrain that, but we still don't understand it that well. And on top of that, now we've got to think about the spectrum of mixotroph strategies and how they're competing with each other. So there's two different kinds of trade-offs in the model. One is what I'm calling a performance trade-off, which is just like how worse are you at doing phototrophy and phagotrophy if you try to do both of them at the same time. And then the second trade-off has to do with like your baseline respiration rate, which may be greater if you're doing phagotrophy. And it seems like both of these trade-offs are probably strong just in order to get like growth curves to look like what our data look like. And so then we have a model where we're just varying irradiance and the input concentration of nitrogen, and then looking at how the trophic strategy evolves. So it's basically like an adaptive dynamics type of model. And what we're finding, consistent with the data, as nutrient supply declines, you get a higher proportion of mixed trophs in the community. And then with light, at least when there's strong trade-offs, under low light, the mixotrophs eventually just get excluded. And then if you look, within the phytoplankton at this continuous trophic strategy, there's a gradient there as well, where the lowest nutrient conditions select for the fastest grazing mixotrophs, relatively speaking. And also you get them under the highest irradiances. But this does depend, so some of this depends on the trade-offs and some of it doesn't. The nutrient effects are very consistent, but the role of light is more complicated. And if you have like weaker performance trade-offs, it can actually go in the opposite direction. I'm not going to show this slide in much detail. But yeah, so I would say, based on these modeling results, if we look across these two gradients, I think nutrient supply is probably the main driver, or at least the most consistent driver, but irradiance may be reinforcing these patterns if the trade-offs are as strong as we think they are. Okay, so to sum up, we've got this phylogenetically diverse group of small phytoplankton, and they seem to be largely organized along this spectrum from autotrophs to like slow grazing mixotrophs, intermediate fast grazing mixotrophs. And that's strongly correlated with where they tend to be most abundant with their niches. And I think probably nutrient supply may be lighter, most likely drivers of that. And I think there's a number of like pretty interesting implications here. If there's fairly consistent shifts in phytoplankton community structure along these major environmental gradients, then that may mean there's shifts in ecosystem processes and food web processes as well, because we know, at least from models, that mixotrophy should alter things like primary production, transfer efficiency up the food web, nutrient cycling, this kind of stuff. Also, because the trophic strategies seem to be varying in a consistent way, pretty strongly with chlorophyll A concentration, we may be able to predict that from remote sensing data, which would be pretty helpful. And of course, the oceans are becoming more stratified and are predicted to continue doing so. So in light of our data, it suggests that the future is going to be more mixotrophic than it currently is. Okay, I'll just mention my few acknowledgements here. Hawaii Ocean Time Series crew collected some samples for us, Terra Oceans. Karen Self helped with some flow cytometry. And big thanks to David for organizing this and the rest of the steering committee. And thanks for your attention.
All right, we have questions for Kyle. There, Suzanne, could you unmute and ask your question? Or maybe...
Yes, can you hear me?
I can hear you.
Awesome. My name is Susanna, and I am a postdoc at the University of Southern California. And this topic talks to my heart, right? Kyle, I love this topic. Great talk. I'm very impressed with the amount of work. And I have many questions. I will try to stick with two of them now. So I was interested on your trade-off, the second trade-off that you showed. that you show that grazing rate would decrease with phototrophic growth rates, right? So I was just interested, for this trade-off, were you including the strictly autotrophic flagellates, or could you observe a trade-off when only looking at the mixotrophic species?
Yeah, that's a good question. So, it's the case both ways. I mean, statistically speaking, there's a significant correlation, even if you just look at the mixotrophs. But there's only 7 of them that we did both experiments on to compare. So more data would be better, but that relationship is there in the data we have. Yeah.
Thank you. And the other question, I was curious about cell size. First, on how much would they vary in cell size if this variation is very tiny among all your mixotrophic flagellates? Or if not, if you could see any correlation between cell size and like grazing capacity and phototrophic capacities among the mixotrophs.
Right. Yeah. So there is some variation. So that chrysophyte that has the highest specific grazing rate is the smallest one. It's like a little bit bigger than 2 microns. Some of the dictyocophytes are maybe like between 4 and 4.5 microns in diameter. So let's say a factor of 2 is the variation that we have. Of course, there's probably larger things that eat Prochlorococcus, like pelagic tunicates, for example. But these are all things that were isolated actually by feeding Prochlorococcus into natural communities and seeing what grew up. So and the size variation is not that big. And and then your question about does that correlate with other traits? Not really that I've seen, no. But the size range is pretty small. Yeah.
Thank you very much.
Stephanie?
Hi, great. Is this on? Yeah. A great talk, an incredible amount of work. I was just wondering, so you had 39 mixotrophs and 4 autotrophs. I was just wondering if that's sort of in that size range, what you think, 2 to 5. I think that's a reasonable percentage of mixotroph or autotroph, or do you think it just came because of the way you selected species?
Right. So, translating the data on these things into real abundances is challenging because there's a lot of biases that arise in the generation of the sequence data. But the autotrophs are a significant fraction of the community based on the metapar coding. Plegomonas in particular at the DCM is consistently found to be quite abundant. And then the percentophytes that we used and other related percentophytes, are consistently seen both at the DCM and in the mixed layer. So, in terms of abundances, it's hard to say, but I would say, they're both there. Maybe the mixotrophs are more abundant than the autotrophs, but it's hard to say how much. In terms of diversity, almost everything is mixotrophic. There's very few things that appear to be autotrophic. And there's some, I put Micromonas in here as autotroph because we don't have any evidence that it's eating things. But then some other people have argued that some Micromonas eat things. It's like a debated topic. So the number of small eukaryotes that are truly autotrophic seems to be pretty small in terms of the diversity. Yeah.
Thank you. Question from Jessica.
Hi, Kyle. Really great talk. I was wondering if you looked at the stoichiometry of the mixotrophs. Do you think that there might be a interaction between the stoichiometry of the prey and the predators?
Right. So we've only looked at the stoichiometry of one of these, unfortunately, which is Florenciella. And that's actually already a paper that's already published. And they have a higher carbon to nitrogen stoichiometry than Prochlorococcus does. I remember that much from that paper. Other than that, I'm not sure. I guess I don't know exactly what you mean by an interaction. Could you be a little more specific?
Well, I wonder if there will be, if they're not getting maybe the kind of nutrients they need, or maybe they're trying to supplement their limiting nutrients with the nutrients in their prey. I know that you just looked at one type of prey, the Prochlorococcus, but I was wondering if if the stoichiometry of the predator would kind of dictate, just what kind of prey or how much phagotrophy they engage in.
Right. Yeah. I guess one thing I can say about that is that bacteria in general tend to be more nutrient rich than phytoplankton do. And where mixotrophs stand, I mean, you might even expect there's some stoichiometric spectrum among these different mixotrophs that we've been studying, but we don't have any data on that. Generally, like autotrophic things or photosynthesizers tend to accumulate carbon relative to heterotrophic ones. And so that might be part of the story as well. But I'm not sure. I can't say much more on how that would affect their need to do phagotrophy, other than that the food they're eating is relatively nutrient rich, probably.
Thank you.
All right. Is there any more questions or any more questions online? Okay. Thank you, Kyle.
Thanks.